Hello and welcome to this short video from the Foundations in Neuroscience playlist. My name is Professor Phil Newton, and in this video we're going to talk about neurotransmitters. What I hope you're able to do by the end of this video then is to be able to define the basic properties of a neurotransmitter system. What are they not, and why are they important? To begin with then, we're going to go back to a video that we saw in a previous video when we talked about action potentials. In that video, we began to introduce some of the key features in the life of a neurotransmitter. We talked about presynaptic and postsynaptic terminals for neurons, and we talked about the release of neurotransmitter from the presynaptic terminal and its actions then on the postsynaptic terminal, or at least some of them. This process is triggered by the opening of voltage-gated calcium channels, and calcium exists on a strong concentration gradient that's more on the outside of the neuron than on the inside. So an action potential travels down the neuron, calcium then floods down its chemical concentration gradient, a series of biochemical steps happen, and neurotransmitter is released from the presynaptic terminal. Those orange blobs there are the big synaptic vesicles that contain the neurotransmitter, and they fuse with the membrane of the presynaptic terminal to cause the release of the neurotransmitter once the voltage-gated calcium channel is open and the calcium that triggers that event has been let into the presynaptic terminal. There are many definitions of what a neurotransmitter is. Almost every neuroscience textbook will talk about defining a neurotransmitter. But for every principle that defines a neurotransmitter, there is also an exception. So we're not really going to go through a text-based list of what a neurotransmitter is. Instead, we're going to take a more pragmatic approach. There are various things that neurotransmitters do, and from a medical perspective, these are probably more important because they are the sites of action of many important medicines. The first of these then is the postsynaptic receptors. These are in the membrane of the postsynaptic terminal, and when the neurotransmitter is released, these are then the site of action, or one of the key sites of action for the neurotransmitter. As you can see by the various different shapes and colors that are used in these in the slide there on the postsynaptic terminal, there is a great heterogeneity within these receptors, these postsynaptic receptors. This can even be on the same neuron. So even though the receptors may all be for the same neurotransmitter, there are going to be different types of receptor. In addition to postsynaptic receptors, then there are also presynaptic receptors. These are also receptors, so they span the neuronal cell membrane. They're for the same neurotransmitter, but they are located on the same terminal, the presynaptic terminal, from which the neurotransmitter is released. The principal function then of these presynaptic receptors, or one of the best understood functions of these presynaptic receptors, is to act as a brake. So once the postsynaptic receptors are all occupied, the presynaptic receptors then, perhaps with a lower affinity for the neurotransmitter, will then act as a brake for the release of further neurotransmitter. A similar function then happens as a result of the transporters, these structures shown in green here. These are basically little vacuum cleaners, often built into the presynaptic terminal, but also elsewhere, that hoover up excess neurotransmitter within the synapse. A similar function is then undertaken by the fourth component in the life of a neurotransmitter. These are digestive enzymes or degrading enzymes. These are normally released by the postsynaptic terminal, and again they serve to mop up excess neurotransmitter to stop it diffusing out and having actions on other terminals. The fifth element in the life of a neurotransmitter that we're going to talk about are the enzymes that make the neurotransmitter in the first place, the synthesizing enzymes. These are normally located in the presynaptic terminal, although they may also be located in glial cells, which we'll talk about in a different video. These non-neuronal cells, which are important for all of the actions at the synapse, including the synthesis of neurotransmitter. The enzymes then often take up the neurotransmitter that's been broken down by the degrading enzymes and reassemble it back into neurotransmitter, pack it up into vesicles so it can be released again. These are the 5 key elements that we're going to go over in a text format. We've got the synthesizing enzymes, the postsynaptic receptors, the presynaptic receptors, the degrading enzymes, and the transporters, all for the same neurotransmitter. All 5 of these can be drug targets. This is a really important principle because many popular science presentations of neurotransmitters will talk about the neurotransmitter itself. You'll often hear about dopamine, for example, being either a pleasure chemical or a love chemical. You'll hear about serotonin being the mood hormone or the happiness neurotransmitter. Whilst there is potentially some basis to some of these statements, what's more important is the actual receptors that the neurotransmitter is binding to. This is where then the drugs themselves that are used to treat psychosis, mood disorders, etc. This is where they have their actions. So dopamine transporter blockers, for example, will increase the synaptic concentration of dopamine, and this can result in a stimulation. This is the way in which drugs like amphetamines work. Drugs which block the transport of serotonin increase the synaptic concentration of serotonin. This is how selective serotonin reuptake inhibitors work. This is also important because these receptors and these transporters and these degrading enzymes, they can be located on the drug targets of the autonomic nervous system, which is again the subject of another video. When the nervous system innervates or controls the function of other organs like the heart or the lungs or the kidneys, drugs that can then be used to treat disorders of those organs actually act on the nervous system innervation of those organs by affecting the enzymes, the receptors, the transporters, and so on. Okay, now we're going to talk about how neurotransmitters are classified. Again, the textbooks have lots of different ways in which neurotransmitters are classified. We're just going to talk about two of them. One of them is classifying neurotransmitters into excitatory and inhibitory. This is a very straightforward but very important distinction between major classes of neurotransmitter, in particular glutamate, which is an excitatory neurotransmitter, and GABA, which is an inhibitory neurotransmitter. Very simply, when an excitatory neurotransmitter is released by the presynaptic neuron onto the postsynaptic neuron, the excitatory neurotransmitter, like glutamate, increases the likelihood that the postsynaptic neuron is going to fire an action potential. An inhibitory neurotransmitter does the opposite. GABA in the human nervous system functions almost exclusively as an inhibitory neurotransmitter, and it decreases then the likelihood of an action potential being fired by the postsynaptic neuron by hyperpolarizing it. Again, we talked about this in the video about action potentials. What I want to emphasize here then is that many neurotransmitters do both, and again the distinction is because of the receptor that it's binding to. The same neurotransmitter can bind to a different receptor, and the receptor can then be inhibitory or excitatory, or as we'll see, modulatory. In order to understand that, then we have to understand a bit more about the biology of neurotransmitter receptors. These distinctions that we're going to talk about applies to both the presynaptic and the postsynaptic receptors that we introduced in an earlier slide. Neurotransmitter receptors can be classified then into ionotropic and metabotropic. Ionotropic are neurotransmitter receptors that conduct ions. They are ion channels. Whereas metabotropic receptors are defined basically by not being ion channels. They do many other things, but they specifically do not conduct ions. There's no pore through which things like calcium or sodium or potassium could travel. The actions of ionotropic receptors are normally faster than those of metabotropic receptors. And ionotropic receptors normally overlap exclusively with postsynaptic receptors, i.e. most of the postsynaptic receptors are ionotropic, most of the presynaptic receptors are metabotropic. Because ionotropic receptors conduct ions, they directly affect membrane potential. Again, referring back to the action potentials video, we talked about the resting membrane potential of about -65, -70 millivolts and how changes in that then a feature of the conduct of an action potential. This is the result of actions at ionotropic receptors. Metabotropic receptors don't directly affect membrane potential. They are normally coupled to intracellular signaling pathways. Those intracellular signaling pathways then can themselves affect ionotropic receptors, i.e. the actions of metabotropic receptors can modulate ionotropic receptors, which then affects membrane potential. But metabotropic receptors themselves do not conduct ions and so do not directly affect membrane potential. Again, a key message to get across is that most neurotransmitters have both ionotropic and metabotropic receptors. If we consider the neurotransmitter serotonin, there are at least 13 different receptors for serotonin. Most of them are metabotropic, but at least one of them is ionotropic. And so the actions of serotonin are going to be very different depending upon the receptors that it's binding to. Different sites in the brain, different parts of the body are going to express different neurotransmitter receptors. This then leads on to a different way to classify neurotransmitters. We've talked about excitatory and inhibitory, and we've talked about the different types of receptors and the way those can be classified. Another way that neurotransmitters can be classified is in terms of their anatomical location. This can be basically broken down into general neurotransmitters that are expressed all over the brain and nervous system, and restricted neurotransmitters whose cell bodies are located in discrete anatomical locations. Generally, when we're talking about general neurotransmitters, we're talking about glutamate, GABA, and glycine. Glutamate and GABA in particular are the start and the stop of the human nervous system. Glutamate is the principal excitatory neurotransmitter, and it makes the nervous system go. GABA is the principal inhibitory neurotransmitter, and it makes neurons hyperpolarize and makes them less likely to fire an action potential. Some restricted neurotransmitters are things like serotonin, dopamine, noradrenaline, and some of the neuropeptides, things like opioids, neuropeptide Y, substance P, and so on. Again, there is an exception to this principle. Acetylcholine is perhaps the most salient of these. In the peripheral nervous system, acetylcholine is a general neurotransmitter, and at the neuromuscular junction, for example, motor neurons release acetylcholine onto muscles and it makes them contract. In the central nervous system, in the brain, acetylcholine is very restricted. There are some discrete acetylcholine nuclei that project them throughout the rest of the brain. And this principle is really important. I'm going to show you it using serotonin. Here then is our favorite sagittal section that you've seen in lots of different videos. I'm going to show you where the serotonin nuclei are located in the brain. They're in these red blobs deep in the brainstem. They're called the Raphe nuclei, and they're very, very restricted. There's just a few discrete locations in which the cell bodies of serotonin neurons are located. However, these serotonin neurons then project their axons all over the brain, all throughout the cortex, into the cerebellum, into the midbrain, into the hippocampus, as you can see by these red tracings here. These are what we would call serotonin projections or serotonergic projections. So the cell body of the neuron is deep in the brainstem in the Raphe nuclei. The serotonin itself is then released in the cortex, in the hippocampus, in the cerebellum, or even further down the brainstem and down the spinal cord. What the serotonin is doing then, what the dopamine might be doing or what the noradrenaline might be doing, is acting as a neuromodulator. And this is another important distinction that maps quite neatly onto this general and anatomically restricted distinction between neurotransmitters. Glutamate and GABA and glycine and acetylcholine in the peripheral nervous system basically function as stop and go signals, what we traditionally think of as neurotransmitters. Serotonin, dopamine, noradrenaline, the peptides, and acetylcholine in the central nervous system, they're normally acting as neuromodulators. They can act as neurotransmitters as well, but we generally think of their actions in terms of a modulatory role. What is a neuromodulator? A neuromodulator normally modulates a neurotransmitter, or it can modulate another neuromodulator. This is getting quite complicated, so I'm going to give you a very simple example of neuromodulation. Opioids are neuromodulators, and one of the things that opioids do, endogenous opioids, our brain makes opioids. One of the things this endogenous opioids does is it modulates the release of another neuromodulator, dopamine. Let me show you how this works. This gives you some example of the level of complexity of the human nervous system and what neurotransmitters and neuromodulators can do. Here then is a cartoon, a schematic of a dopamine neuron. You have dopamine neurons deep in your midbrain, and they are firing what we call tonically. So they're constantly ticking over, firing away and releasing dopamine. Changes in the firing rate of these dopamine neurons are important because they can signal, for example, the availability of unexpected rewards or the lack of a delivery of a reward that was expected. When we get a reward that was unexpected, the firing rate of these dopamine neurons goes up. When we are expecting to get a reward and that reward is not delivered, then the firing rate of these dopamine neurons goes down. And by reward, we mean a biological reward, for example, the availability of a palatable food or a mate or something similar. So this dopamine neuron is ticking away, it's releasing dopamine. You've got DA there, the cell body of the neuron, and you've got then the little molecules of DA, the smaller circles, those are molecules of dopamine being released as the neuron fires away. The release of dopamine from these neurons is then controlled in part by the action of GABA on the cell body of the dopamine neurons. Remember, GABA is an inhibitory neurotransmitter. So when GABA binds to the cell body of the dopamine neurons, it hyperpolarizes these dopamine neurons, makes them less likely to fire an action potential, so turns down the firing rate of these dopamine neurons, and less dopamine is released. Now, the actions of GABA are themselves modulated by the neuromodulators opioids, endogenous opioids. So our brain makes opioids as neuromodulators. These GABAergic interneurons, they have receptors for opioids, and the actions of the opioids when they're released by these opioidergic neurons is that it hyperpolarizes the presynaptic terminal of the GABA neuron. So the GABA neuron is then less likely to fire an action potential, and so it is less likely to release GABA onto the dopamine neuron. Remember, the GABA was acting as a brake on the firing rate of the dopamine neuron. When the GABA is then less likely to be released, the brake is removed, and so the firing rate of this dopamine neuron can go up, and more dopamine can be released. This is part of the cellular mechanism by which our brain's reward system responds to unexpected rewards. There is a burst of opioid release. This then turns off the brake on the dopamine neurons, and this causes an increase in the release of dopamine due to an increase in the firing rate of these dopamine neurons. In this way, the opioid is acting as a neuromodulator by modulating the release of GABA. The GABA is then modulating the release or the firing rate of dopamine, and so we can turn up or down the firing rate of the dopamine neurons. There are also GABAergic projections, which themselves release GABA onto the GABAergic neuron. This, just like the opioid, is a double negative that can reduce the release of GABA from the interneuron onto the dopamine neuron, and again can turn up the firing rate of the dopamine neuron. The summary then of everything we've covered in this video: there are multiple components to a neurotransmitter system. Receptors, transporters, breakdown, and synthesis enzymes. All of these are drug targets, and they're more specific than the neurotransmitter itself. So they're actually more important when trying to understand the pharmaceutical and so the medical clinical applications of neurotransmitter biology. Neuromodulators are normally more anatomically restricted, and they modulate the function of neurotransmitters. For example, GABA and glutamate, and then GABA and glutamate can then modulate the release of other neurotransmitters, as we saw in the example with dopamine. That's it. Any questions, please do send me an email, and I'll see you in another video. Bye-bye.